Good evening, participants. So we are going to start bioinformatics course from today. So first of all, please subscribe my channel, YouTube channel. Please all subscribe my channel. After that, we will go ahead. Okay. My voice is clear to all. Please comment if my voice is clear. Good evening, participants. Okay, good. So you are more comfortable in English or Hindi? Please comment. Okay, no problem. Little low. I will speak loudly. Okay. Okay, no problem. English both and others, please. Okay. So I am Dr. Anand Kumar. I've completed my PhD from IIT BHU. Currently working as an assistant professor of biotech. Okay. So here we are going to discuss about the bioinformatics course. First of all, uh, you will introduce yourself. Uh, okay. Okay, not today because you cannot speak here. Okay, so tomorrow I'll take introduction. Okay, so today we will start basics of bioinformatics, just introductory class. Okay, so because in a live YouTube live studio this is not possible. Okay, so for the introduction we will use Zoom class. Okay, so we are going to start uh, basics of bioinformatics. So I think my screen will be visible to all. Okay, so here today we will discuss about the introduction of bioinformatics and historical overview of bioinformatics. After that we will go for the human genome project. After that biological databases. After that internet and bioinformatics, and knowledge discovery and data mining. In the last, we will discuss about the career prospect in bioinformatics. Okay, so we will cover all these topics here. Okay? So we are going to discuss about the introduction of bioinformatics. Okay, so first we will talk about the definition of bioinformatics. A computational approach solves the biological problem. Okay, this may be a definition of bioinformatics. Okay, next definition may be a bioinformatics is emerging and advanced branch of biological science, contain biology, mathematics, and computer science. Actually, it involves bioinformatics involves three branches. Okay, that are the biology, mathematics, and computer science. This is a combination of biology, mathematics, and computer science, right? So, next definition may be bioinformatics developed a new thought to maintain the concepts and store the huge amount of biological data. Okay. Next stage, bioinformatics concepts and method are different than that biological concept and method. Okay. Next, bioinformatics are logical and technical means by which not only solve the biological problems but also can predict the new aspect. Okay. We have learned about the definitions of bioinformatics and basics bioinformatics. Okay, so here we are going to discuss areas of bioinformatics. So just you are a student of biotech and basic science, life science, bioscience. Okay, whatever you have. So here we will talk about the uh, related areas like genomics, proteomics. Okay, computational languages. This is a part of computer science. Okay, here we will study about the C++, Perl, BioPerl, BioJava. These are the programmings. Okay, so you can learn these languages under the computer science course. Okay, so and here bioinformatics also cover biostatistics. Okay, and some more branches like computational biology, database management system. Okay, uh, systematic biology, and chemoinformatics. Okay, all these are the branches of bioinformatics, so we can go ahead for this study. Now, uh, computational biology means in silico areas of bioinformatics. So main thing is computational biology. In computational biology, we can study about the comparative homology modeling and phylogenetic analysis, protein structure prediction, protein folding problem, microarray analysis, docking approaches, and new drug discovery, right? So in a comparative homology modeling, we will go for the if you have a amino acid sequence of different peptides. Okay, so you can compare a sequence, and if you have already a, 3D structure you have determined through uh, NMR and X-ray crystallography, so you can compare uh, your with your amino acid sequence. If this is similar to other, and other one have been determined the 3D structure, so your amino acid sequence may form a similar structure. Okay, and your proteins maybe have a similar functions, right? So in the same thing, we can go for the phylogenetic analysis. Okay, you can study for the evolutionary history and You can also determine predict your protein structure and protein folding and microarray analysis for the detection, and after that docking. Okay, so these are the all branches of computational biology. Okay, now we are going to discuss historical overview of bioinformatics, right? So all these things you have to remember. Okay? In 1859, the on the origin of species, that is the book published by Charles Darwin, that introduced theory of genetic evolution. Okay, allow adaptation over time to produce organism best suited to the environment. Okay, so you have to remember here, time means year, and name of the book, and author means Charles Darwin. Okay, after that in 1869, the DNA from nuclei of white blood cells was first isolated by Frederick Mischer. Okay, he called nuclein. Okay, now it is called, we are calling it nucleic acid. Okay, after that in 1951, Linus Pauling and Corey proposed the structure for the alpha helix and beta sheet. Okay, after that in 1953, Watson and Crick proposed the double helix model for DNA based on X-ray data obtained by Franklin and Wilkins. Okay. In 1955, the sequence of first protein to be analyzed, bovine insulin, is announced by F. Sanger, Frederick Sanger. Okay, so this is the first insulin sequence. Okay, you have to remember, this is also a recombinant protein, first recombinant protein produced by genetic engineering. Okay, in 1955. Okay, next one is the 1958, the Advanced Research Project Agency, ARPA, is formed in the US. Okay, after that 1973, the Brookhaven Protein Data Bank, okay, is announced. This is the protein data bank. All the amino acid sequence and 3D structure have been submitted in this protein data bank. Okay, okay, if you are characterizing your protein, and after determining structure or sequencing of any amino any peptide, you can submit your data in the databases. Okay, in 1987, Perl Practical Extraction Report Language is released by Larry Wall. Okay, you know about the Perl. This is a language. Okay, computer programming language. Okay, after that in 1988, National Center for Biotechnology Information, we are calling it NCBI in short, founded at the NIH and NLM. Okay, National Institute of Health. Okay, in 1990, Human Genome Project was launched. Okay, and BLAST program introduced by S. Carlin and S.F. Altschul. Okay. Tim Berners-Lee, a British scientist, invented the World Wide Web in 1990. Next, in 1992, the Institute for Genome Research, associated with plans to exploit sequencing commercially through gene identification and drug discovery, was formed. Okay. Uh, in 2001, the human genome approximately 3000 megabase pair was published. Okay, you are assuming it is how long? 3000 megabase pair. Next, future goals of molecular biology and bioinformatics research. Actually, we cannot uh, go ahead in a molecular biology field without bioinformatics. Actually, molecular biology and bioinformatics both are the intact field. Okay, so in 2010 completion of the 2010 project to understand the function of all genes within their cellular organism and evolutionary context of Arabidopsis thaliana. Okay, but we are expecting that in 2050 to complete of the first computational model of the complete cell, or maybe even already a complete organism, right? So I hope you are understanding whatever we are discussing here. Please tell me you are getting whatever I am discussing here. Please, my voice is audible to all, right? Please comment. So if you have not subscribed my channel, YouTube channel, please subscribe first. Okay, please subscribe. If you are not subscribed my channel, please subscribe. Okay, now everyone is getting whatever we are discussing here. Okay, now we are going to discuss about the human genome project. Okay, so US government project coordinated by the Department of Energy and National Institute of Health, launched in 1986 by Charles Delacy. Okay, actually, first of all, we want to know about the genome. What is genome? The whole hereditary information of an organism that is encoded in the DNA. This is known as genome. Okay, and what is the aim of this project? So, to identify the approximately 1 lakh genes in the human genome. Okay, approximately 1 lakh genes are present in the human DNA. Okay, so how we can identify, how we can characterize all the genes? This is a big task. Okay, and to determine the sequences of the 3 billion bases that make up human DNA. Okay, our human DNA is of approximately 3 billion bases. Okay, this is a very big, very large. Okay, to store this information in databases. Okay, to develop tools for the data analysis and address the ethical, legal, and social issues that arise from genome research. Okay, because this is not ethically right to clone any to clone of human. If we have sequenced the human genome, that we can develop clone. Okay, so this is ethically wrong. Okay, so you can see here the sequence of human mitochondrial DNA. Okay, this is the sequence you are seeing here in a picture. So a group of researchers have managed to complete a genetic map of the bacterium Haemophilus influenza that was firstly sequenced. Okay, the approach called whole genome shotgun sequencing to sequence the 1749 genes of the bacterium in a minimum time period. Okay, the next is the Haemophilus influenza project was based on an approach to genomic analysis using sequencing and assembly of unselected pieces of DNA from the whole chromosome, right? Now, next we are going to discuss about the benefits of human genome project research. Okay, so improvements in medicine and drugs used in genetic or metabolic disorder. Okay, next one is the microbial genome research for biofuel and environmental cleanup. Next one is DNA fingerprinting and forensics. Next one is improved agriculture by improving the wild gene of high yielding variety of grain and livestock. Okay, next one is the better understanding of species evolution and human genome. And next one is more accurate risk assessment by gene mapping. Okay, so these are the benefits of human genome project research. Now we are going to discuss about the internet and bioinformatics. This is the part of computer science. Okay, so we will learn about the basics of computer science here. Okay, so internet plays an important role to retrieve the biological information, right? Next, bioinformatics. Emerging new dimension of biological science include the computer science, mathematics and life science. I already told you this thing. Next, the computational part of bioinformatics used to optimize the biological problems like metabolic disorder, genetic disorder. Okay, so computational part contains software and tools development. Second thing, database development. Third thing, software and tools applications. Okay, fourth one is the operating system, Windows 2000, XP, Linux, Unix. Okay, like that. So here are some more information. The mathematical portion helps to understand the algorithm used in bioinformatics software and tools. Okay, the mathematical portion which used in bioinformatics are matrices, biostatistics and complex mathematics functions, differentiation, integration. Okay, all these are involved in the bioinformatics. These all are the parts of mathematics approach. Okay, so there are different types of matrices for the sequence alignment, blast, FASTA, MSA and phylogenetic prediction. Okay, for the biostatistics, HMM, ANN in secondary structure prediction, artificial neural networking. Okay, so in the next next, differentiation and integration, time and space complexity, E value, P value in BLAST. We have to consider all these parameters. Okay. So next is the internet is a global data communication system. It is a hardware and software infrastructure that provides connectivity between computers. The web is one of the services communicated via the internet. It is a collection of interconnected documents and other resources linked by hyperlink and URL. Okay, so you are seeing in a picture the portal interface. Okay, so different types of browsers, web service. Okay, means first manufacturing information, employee data, supply, inventory, customer record, product data. All these will be analyzed and will go for the web internet application portal, and all these will be analyzed. Okay, so this is the theoretical part. Okay, after that we will discuss uh, about the practical part. Okay, so you have a laptop. How many of you have a laptop? Without a laptop, practical sessions are not possible. Yes, I'll share with you. How many of you have a laptop? Without laptop, a practical session will be very difficult, because some softwares will not run in a mobile phone. Please comment in a comment box. So if you not have, please arrange laptop. Okay, so some other internet resources for bioinformatics are database search engine like NCBI, Swissprot, Uniprot K. Okay, next one is online serve tool, cluster W, Swiss model server. Next, rational database management system. Next one is the other software and information resources. Okay, and the next one is online databases, KEGG, Modbase, PDB, Zinc database, Mole software, and other one biological databases, uh, primary, secondary, and composite. Okay. Next one is bioinformatics softwares application, bioinformatics softwares development. Now we are going to discuss about the biological databases. Okay, so there are different types of biological databases. uh, Bibliographic database for the literature, taxonomic databases for the classification, nucleic acid databases for DNA information, and genomic databases, gene level information, protein databases for protein information, protein families, domains, and functional sites for the classification of proteins and identifying domains. Okay, the next one is the enzymes metabolic pathway, to know about the metabolic pathway. Okay, so all these are the biological databases. So here now types of biological databases accessible. Okay, so there are three basic databases: primary database, secondary, and composite. Okay, so In a primary database for nucleic acid and protein, we will use EMBL, European Molecular Biology Laboratory, and Gene Bank, and DDBJ, DNA Data Bank of Japan. Next one is the Swiss Prot, Tremble, and PIR. Okay, so all these are the primary databases. So in a secondary databases, we will use ProCite, PFAM, blocks, prints. Okay, so next one is the composite databases. Okay, so in a composite databases, NRDB, natural resource database, and OWOL, wave ontology language. So there are basically three databases which are in collaboration, international collaboration. These are the DDBJ, EMBL, and Gene Bank. Okay, DNA Data Bank of Japan, European Molecular Biology Laboratory, and Gene Bank. Okay, meaning of that this is that um, if you are submitting any sequence, means nucleic acid sequence or protein sequence, and in a database like DDBJ, you can search from anywhere in a EMBL or Gene Bank. Okay, this will be available in any database. Okay. So Gene Bank is the NIH genetic sequence database, an annotated collection of all publicly available DNA sequence. Next is the DDBJ, DNA Data Bank of Japan, began. हेलो, हाँ सर बताइए. क्लास के बारे में. नहीं मतलब क्लास चल रहा है सर. नहीं नहीं नहीं, बायोइन्फॉर्मेटिक्स. हाँ हाँ सर, क्लास चल रहा है. आप देखिए सर, लिंक मैंने भेजा हुआ है. ठीक है, मैं चेक करता हूँ. ठीक है, मैं चेक करता हूँ. ओके ओके. प्लीज वेट फॉर अ मिनट. ओके, आई विल कंटिन्यू. coming back on DDBJ. Okay, so DNA Data Bank of Japan began data bank activities on in earnest in 1986. Okay, at the National Institute of Genetics (NIG) with the endorsement of Ministry of Education, Science, Sport and Culture. Okay. Next one, the Center for Information Biology at NIG was recognized as the center. Hello, hello, hello. हाँ जी. So the Center for Information Biology at NIG was recognized as the Center for Information Biology and DNA Data Bank of Japan, okay, in 2001. The new center is to play a major role in carrying out research in information biology and to run DDBJ operation in the world. Hello, hello. Good evening. भैया अभी थोड़ी देर में बोलना बात करना ठीक है अभी. हाँ ठीक है. Next one is the EMBL. Okay, EMBL nucleotide sequence database. The EMBL nucleotide sequence database, also known as EMBL bank, constitutes Europe's primary nucleotide sequence resource. Okay, main sources for DNA and RNA sequences are direct submission from individual researchers, genome sequencing projects, and patent applications. Okay, so the database is produced in an international collaboration with Gene Bank USA and the DNA database of Japan DDBJ. Right now, knowledge discovery and data mining. This is the part of computer science and mathematics. Okay, so I'll share these PPT with you, so you have to just get a brief knowledge, so you can read and basics of biology like what is gene and what is genome and like this. So you know very well if you are the student of biotech and bioscience, life science like that. Okay, so next you can read about the data mining. Okay, so I'm not going to discuss in a detail here. I'll discuss just in a brief. Okay, so a gene can be identified as a region of DNA. A genome is one haploid set of chromosomes with the genes that contain. Perform competent comparison of gene sequences across species and account for inherently noisy biological sequences due to random variability amplified by evolution. Okay. Assumption: if a gene has high similarity to another gene, then they perform the same function. Okay, so next is the analysis, language and goals. Feature is an extractable attribute or measurement. Gene expression, location, pattern recognition is trying to characterize data pattern. For example, similar gene expression, equidistant gene locations. Data mining is about uncovering patterns, anomalies, and statistically significant structures in data. For example, find two similar gene expression with confidence greater than X. Okay. So, data mining, knowledge discovery from data, extraction of interesting, non-trivial, implicit, previously unknown, and potentially useful patterns or knowledge from huge amount of data. Data mining, a misnomer. Alternative names. This is the alternative name. knowledge discovery mining in databases KDD, knowledge extraction, data pattern analysis, data archaeology, data dredging, information harvesting, business intelligence, etc. Watch out is everything data mining, simple research, simple search and query processing, deductive expert system. Okay, so this is the evolution of database technology. Okay, so just you have to get a basic idea about this. Okay, not in detail. So why data mining is important and what are the potential applications of data mining, right? So you are seeing here the architecture, typical data mining system. So this is the knowledge discovery process. Actually, we are not go uh, will not go here in a detail, just in a brief. Data mining core of knowledge discovery process, like you are seeing here the figure. Okay, data cleaning, data integration, selection, data mining, pattern evolution and knowledge. Okay, so last part is the career prospect in bioinformatics. Okay, so here, mm, you can develop your career as a scientist, research associate, or you can develop your career by uh, be a professor, reader, lecturer in university or college, or technical executive in business and development, data analyst in scientific organization and lab, biosoftware development in IT industry. Okay, so these are the fields in which you can develop your career. Okay, so this was the basics of bioinformatics. So uh, most of the things in this session. So please tell me if you got uh, something new. Please comment. Please comment. Please comment. So first we will discuss here about the different types of databases like NCBI, how you will search a sequence, nucleic acid sequence and protein sequence. Okay, we will discuss about these things like BLAST. Okay, and after that we will go for the proteomics study. Okay, in a proteomics we will go for the homology modeling. Okay, structure prediction, like that. And before that we will learn about the how to design primers using different types of softwares like gene tool light software. Okay, so how you can maintain a reading frame for the expression. I am again saying that please subscribe my channel if you have not subscribed yet. Please subscribe. Please subscribe channel first. I am seeing here only 3 to 4 people have subscribed channel, not more than that. Okay, so please subscribe channel first. So after that we will start. Tomorrow we will discuss about the internship program. Okay, so I'll tell you how to write a review paper. Okay, what is the structure of review paper. I'll share different types of topics on which you can uh, go for the writing of review paper. Okay, so you will get the basic idea about the review paper. So today we will finish our session here. Okay, so please I am saying again and again, please subscribe channel. Okay, so that you can get that video whatever I am discussing here, you will get the link. Please subscribe. So thank you very much for today. I hope you learn something new, and I'll try to deliver uh, something new and important for you. Okay, for the bioinformatics. Okay, thank you very much for today's session.